We're live.
Thank you. Good afternoon. My name is Shantae Hill Hammond, and I am the designated federal officer for the Science Advisory Board Biosolids panel. In my role as DFO, I would like to formally open this public meeting and share some opening administrative remarks. The SAB is an independent, expert federal advisory committee chartered under the authority of the Federal Advisory Committee Act, also known as FACA. The SAB is empowered by the Environmental Research, Development, and Demonstration Authorization Act, also known as ERDA, to provide advice to the EPA administrator on the scientific and technical underpinnings of EPA's decisions. This panel and all committees of the SAB provide advice through the chartered board. FACA and EPA policy require that SAB meetings be announced to the public in the Federal Register, and that substantive deliberations and interactions with EPA and the public be conducted in open sessions where a DFO is present to ensure the requirements of FACA are met. FACA also requires that public meetings provide an opportunity for oral and written public comment. There is a public comment period noted on the agenda for members of the public who have registered in advance with the SAB staff office to present oral comments for this meeting. No members of the public have registered to provide oral comments today. Members of the public have provided written comments, and those have been posted to the SAB website as well as distributed to the panel members. The SAB consists of special government employees appointed by EPA to their positions. As government employees, all members are subject to applicable ethics laws and implementation regulations. The SAB staff office has determined that advisors participating in this meeting have no financial conflicts of interest or appearance of lack of impartiality under ethics regulations specified in the Code of Federal Regulations at Title 5, Part 2635, relating to the topic of this meeting. This is the first of three planned meetings where the panel will conduct a peer review of EPA's draft Biosolid Risk Assessment Framework. All meeting dates were announced in the Federal Register in a notice published on Tuesday, March 14th, 2023. For today's meeting, the panel will receive a briefing from EPA and review charge questions. Panel attendance has been taken and we do have a quorum for today's meeting. Again, the meeting is open to the public, and the public may access this meeting by telephone or by webcast. To access the webcast link, please see the SAB webpage at epa.gov/SAB, select upcoming meetings, and then the link for today's meeting dates. Please note that all meeting materials, including the agenda, agency presentations, and public comments, are posted on the SAB website. As members of the public are joining and participating in this meeting, I will ask that all speakers identify themselves prior to providing remarks. And if you are not speaking, please ensure that your microphones are muted. Minutes will be prepared and certified as accurate by the channel pair to summarize discussions and action items in accordance with the requirements of FACA, and minutes will be available on the SAB website. Finally, all participants are asked to acknowledge their attendance at today's meeting by providing your name and affiliation to me by email. My email address is hill-hammond.shante@epa.gov. Please note that my email address is also on the SAB webpage for today's meeting. Before I turn the meeting over to our chair, Dr. Sylvie Broder, I would like to introduce Mr. Tom Brennan, Director of the SAB Staff Office, and also Ms. Colleen Flatherly, Acting Director with the Office of Water, to provide welcoming remarks. Tom?
You're muted.
Okay, let's try again. So thank you and welcome everyone to this public meeting of the SAB Bioscience panel. I'd like to first thank the team from the Office of Water for all the hard work they did to make today possible. They've done a lot of work on this tool and I'm really happy to be conducting this peer review. I'd also like to thank my teammates in the SAB staff office and our contractor, Indina and Sai, for all the support it takes to run a meeting like this. With a special thank you to Shantae Hill-Hammond for being our designated federal officer. It's a lot of work, and thank you, Shantae. Next, I'd like to thank all the panel members for taking time out of your professional lives to provide this public service to the agency and to the country. I really do appreciate the effort that you took to make yourselves qualified for this panel, to prepare, and the peer review you're about to conduct. So thank you all. With a special thanks to our chair, Dr. Sylvie Broder. Sylvie, it's a lot of work to be a chair, and I really appreciate you stepping in and taking a leadership role. And now I'd like to turn it over to Colleen Faraday from the Office of Water for her opening remarks. Thank you, Colleen.
Thank you, Tom. Good afternoon. My name is Colleen Flaherty, and I'm currently serving as the Acting Director of the Health and Ecological Criteria Division in EPA's Office of Water. First, I'd like to also thank the members of the SAB panel for taking time out of their busy schedules to review and provide feedback on EPA's proposed risk assessment framework for contaminants found in biosolids. This effort is central to fulfilling EPA's statutory requirements under the Clean Water Act for use and disposal of biosolids. We also appreciate input from the public and look forward to receiving comments from all stakeholders on these activities. I'd like to emphasize certain aspects of the materials we provided for review to ensure that the SAB and stakeholders understand what EPA needs from the SAB. EPA is required under Section 405 of the Clean Water Act to assess the potential human health and ecological risks posed by contaminants found in biosolids. In doing so, the agency has to consider factors relevant to exposure and toxicity of contaminants in biosolids. Our overarching goal is to create a framework to prioritize and assess the potential risks of over 700 chemicals that have been identified in biosolids. We are proposing a three-step process to do so. First, we want to prioritize chemical pollutants reported in biosolids for risk assessments. Second, we want to conduct screening level risk assessments using a customized model for this purpose. And third, we want to conduct more refined risk assessments for chemicals that pose the greatest risk to human health or the environment. I'd like to make a couple of important points about the desired scope of this SAB review. The first point is that this review is not meant to focus on any particular chemical or set of chemicals. Our charge questions are clear that we are asking the SAB to focus on the general three-step process that I just described and its applicability to over 700 chemicals found in biosolids. We have included some illustrative examples using specific chemicals to demonstrate how we intend the framework to be used. However, these specific chemicals should not be the focus of this SAB review. That said, it is possible that some of the SAB feedback may be informative for specific biosolids risk assessments in the future. The second point I'd like to make is that this review is not meant to focus on any specific models underlying the framework. We ask that the SAB focus on the construct of the proposed framework and the appropriateness of the models for evaluating human health and environmental exposures related to biosolids use or disposal. All of the underlying models have been externally peer reviewed. We are not asking for additional peer review of the underlying models, but rather whether they are appropriate for biosolids risk assessment as proposed. Thank you again for taking time to participate in this important peer review process, and we look forward to your feedback.
Thank you for your comments, both Colleen and Tom Brennan. And with that, we'll turn the meeting over to our chair, Dr. Broder.
Thank you, Shantae. I believe you want me to take the roll call officially, right? Okay, I'm going to start by taking the roll call for the panel. And so just unmute yourself and quickly say here when it gets to you. So, and I apologize if I misstate your name, but Florence Honoru.
Here.
Thank you. Jennifer Arp.
Here.
Lisa Camp or Campy?
Camp. Here.
Paul DeLeo.
Here.
Greg Kester.
Here.
Linda Lee.
Here.
Drew, I think, is late and I don't see him yet. Michael McFarland.
Here.
Ian Pepper.
Present.
Jennifer Samel.
Yes, I'm here. Jennifer Sommel. Thank you.
Okay, thank you. All right, thank you everybody for being willing to serve. This is not a little task and it is certainly important. The agenda for today, you've received it, but just a brief overview. We're going to receive 2 presentations from the EPA. We're currently scheduled to have a break in between. And then given that there are no public comments, we'll go into a review of the charge questions themselves. We'll go through question by question, allowing you to ask questions. I guess the public's comments are after going over the questions. Then we'll move into the next steps and we'll adjourn. The times on the agenda are approximate and some things may take longer than others, and that's fine. The point of the gathering is to make sure that we get our questions, the panel's questions regarding the charge questions, addressed before we move into working on the charge questions. And having said that, so we'll take as long as we need for each topic and then we'll move to the next one. And with that, I'm going to turn it over to Richard Judson for the first EPA presentation.
Okay, thank you. Let's go to the next slide, please. Okay, so the goal of this presentation is to describe the overall prioritization framework. So hopefully that'll be clear. You've heard some of these points before. So the Clean Water Act requires Office of Water to evaluate chemicals that occur in biosolids and focusing on both human health and environmental species. And the sewage sludge surveys and literature surveys have found over 700 chemicals that are in the biosolids. And OW has developed a screening tool for doing the screening level risk assessment we already heard about, but it would be very difficult to put all 700 chemicals through that all at once. And so there's a need to prioritize which chemicals go in first. And so this, what we call the public information curation and synthesis, and I'll only say that once, the PICS process is being proposed to use to prioritize what goes into the screening level risk assessments. And the PICS process was initially developed to help with TSCA, the Toxic Substances Control Act, to prioritize chemicals. And turns out the rationale or the pieces that you want to use for prioritization are very, very similar between biosolids and TSCA. So we believe it can be used more or less as is, but I will talk about some tweaks we may do. So next slide, please. So in the TSCA context, which is where we developed this initially, going back down to the 3rd bullet point or the 4th bullet point, under the new Frank-Lautenberg Act for TSCA, which regulates new and existing industrial chemicals. Periodically, and it's essentially every year, the OPPT needs to designate 5 chemicals or 10 chemicals as high risk, 5 as low risk, and the high risk chemicals have to go through full-blown risk assessments. And there are 30-odd thousand chemicals on the TSCA inventory. And so clearly it would be very difficult to go through all 30,000 by hand and figure out which ones to put into that tin this year, into next year, and so on. So that was the origin of the PICS process. Next slide, please. So we thought about very hard what PICS was supposed to do and what not. And there's a lot of words here and I won't go through everything, but we're using public data. So for TSCA and for biosolids, we're going to be using public data. We don't have access to, or we don't want to bring in CBI data, because we want this to be a very transparent and reproducible process. So the process I will describe is computational. So there's some computer databases, computer software that implements some rules and some scoring schemes. And so if you want to change the rules, you can change that, but every time you run it with a given set of rules, it runs the same way. And anyone can go in and look at how the rules are defined and implemented. So it's very transparent and it's reproducible. And what this means is that the manual work one does under either TSCA or Biosolids can really be focused on the high priority chemicals and not looking through all of the data for 700 or 30,000 chemicals. Another feature of the PICS is that it's very flexible. It's modular. So we have modules for human hazard, for ecological hazard, for fate and transport, and so on. And those can be modified as appropriate for the particular domain. What it's not supposed to do, we don't want to create a ranked list of substances saying this is the most worrisome substance and this is the least worrisome substance. We don't want to signal because something comes up to the top of the list, and I'll show you what that means later on, that EPA automatically has concerns about that particular substance. This is really a very initial process of prioritizing, and as the risk assessors dig into the data, they will maybe not use some of the data we used in PICS because it doesn't meet quality, or they may go do further literature searching that would bring in data PICS does not have. And so it's not supplanting expert judgment and review. It's not using CBI data. And we're not doing systematic review up front. It really is a very kind of quick and dirty look at all of the public data we can pull together and say which chemicals at first glance look high concern, low concern. Next slide, please. So the overall process is broken into three main pieces. And the first piece is data QC. So when we started on the TSCA process 3 or 4 years ago, we wanted to know how good was the data that we're pulling from these public sources. And so we developed a QA plan and we looked at resources and we developed some QC tools. And the major point I want to say about this, and go on to the next slide, please, is that to do full manual QC on every data point, which is hundreds of thousands of data points, you simply couldn't do. But we have subsequently looked at, developed some tools to more automatedly do QC on data, and where chemical records are flagged to do actual manual QC. But one of the key points here is this first major bullet point, what we call type 1 data sources. They are data that's easily pulled in. And as example, EPA, so the European Chemicals Agency, has a big database of chemical studies that registrants in Europe have submitted. We can pull that in easily. So that'd be a type 1 data source. The non-type 1 would be doing a full-blown literature survey. So we are not, at this point, doing a full literature survey on either the TSCA chemicals or the biosolids chemicals. That would be a second tier in the screening level risk assessment or the full risk assessment. I think the major point about the second bullet point is we've learned a lot about QC since we did the TSCA, and as appropriate, we can describe that in more detail later on. Next slide, please. Okay, so a lot of the detail I'll describe here, which is TSCA relevant, but the themes are all the same. We looked at 238 chemicals, which is same order of magnitude as the 700, and it's different from the 30,000. So I'll show some results on this particular 238 chemicals. But we think about the data lying along two axes, what we call the scientific domain metric, which is really a surrogate for how risky the chemical is. Is it a high risk chemical or a low risk chemical? And the other is information availability metric, how much the data that you would want to have to do a full-blown risk assessment do you have? Because you don't want to go into a risk assessment where there's likelihood you don't have key data available. You don't have studies on aquatic species, for instance. And so we want to look at both of those two things together. Next slide, please. So the scientific domain metric is broken into these, I believe it's 7 categories. Human, and I'll go into those a little more detail. Human health to exposure ratio. So it's really a margin of exposure. Carcinogenicity, genotoxicity, ecological hazard, susceptible populations, persistence and bioaccumulation, so fate and transport, and skin sensitization and eye irritation. And then on the information availability, we look at relevant kinds of studies and information. And depending on some properties of the chemical, we may not require all of those. And I'll go into more detail later. And then what you finally put together is a little picture, and I'll show this picture several times, which is a graph with the information available. That's a hard word for me to say. Information availability metric on the x-axis, scientific domain metric on the y-axis, and the chemicals in the top right would be the ones we would probably prioritize for further risk assessment. We have good information coverage, and they look to be of high concern. Next slide. Okay, so the reason we chose these specific 7 scientific domains was because they were previously used under TSCA. There's some statutory language. We worked a lot with the program staff. But having said that, in discussions with the Biosolids group, they are pretty much all relevant to the Biosolids use case, and it's not clear that there are any specific domains we would add it, with one exception, and I will get to that at the appropriate time. And so for each of these 7 domains, we have a tiered workflow, which I'll describe one example of. And then, so we have essentially 7 values, one value for each of these domains, and you simply add them up to get the overall scientific domain metric. An important point, which is something you might consider as you opine on what we're doing here, is the domains are all equally weighted. Human health to exposure ratio gets the same weight as genotoxicity. Okay, maybe for biosolids, it might make sense to change the weights around somewhat. Next slide, please. So go into a little more detail on the 7 domains. So there's the human hazard to exposure ratio, the margin of exposure. And so what we start off doing is taking points of departure from in vivo studies. If we have that, that's what we would use. If we don't have in vivo studies, we would take in vitro, and we do in vitro to in vivo extrapolation. And if we don't have that, we can use what's called TTC, threshold of toxicological concern. And each of these would give us a point of departure in mg per kg per day. So we focused on oral exposures for the TSCA, and I believe that's going to be the same for the biosolids. We then used a generic exposure model of models called SEAM3 in the TSCA use case. Here, the biosolids has a biosolid specific exposure model that we would use instead. The next domain is carcinogenicity, and these are definitive cancer calls from, for instance, IARC or the Office of Pesticide Programs. Genotoxicity is, again, definitive calls. This chemical is or is not genotoxic based on extensive data. Ecological side, we did not have an exposure model for ecological species, aquatic species, at the time we did this. That is being remedied. And so we will probably, for biosolids, and actually use the hazard to exposure ratio here also. We look at vertebrates, invertebrates, plants, and we look at short and long-term studies. And for the most part, we used in vivo studies, but if we don't, there's some acceptable QSAR models which give points of departure for aquatic species. In susceptible populations, for TSCA, the only susceptible population we looked at was children exposure. So what's the likelihood children will be exposed to this particular chemical? And we're discussing other populations we might add into the biosolids situation. Then we look at persistence and bioaccumulation and skin sensitization and skin and eye irritation. There's a little bit of discussion about whether this was biosolids relevant, but it was concluded it is, because if you have workers in waste treatment plants who are exposed to chemicals that could sensitize them, that's worrisome, or farm workers who are exposed, when the biosolids are spread onto farm fields. Next slide, please. So this is an example of the workflow. And so for each of the 7 domains, we have a workflow like this. And just a standard decision tree. So does the chemical have an exposure estimate? If it doesn't, we set it aside and say that, we can't give you a value for this domain. And we have one of these IG as information gathering. We set an information gathering flag here, which says, okay, you need to go look at that. Assuming we do have an exposure estimate, we say, do we have an in vivo repeat dose point of departure? And if so, then we go ahead and divide that by the exposure estimate, and we calculate the HER, the hazard to exposure ratio. If we don't have an in vivo value, we do the in vitro to in vivo extrapolation using these new approach methods. And we calculate what's called a bioactivity to exposure ratio, a BER, which is the same units. And then finally, if we don't have that, we calculate this threshold of toxicological concern. We remove, there's a 100x safety factor, which is not relevant in this use case, and we calculate the TTC to exposure ratio, the TER. And then that goes into this to calculate the hazard metric. And we'll talk about that the next slide. And if you can't do either of those, either in vivo, in vitro to in vivo, or TTC, then you again set this information gathering flag, and you get penalized later on for that. Next slide, please. And this is a table from the TSCA report, which I believe that or its version for biosolids you have. But for every one of the 7 domains, we give a score of 0 to 4. And a 0 is we don't have the data to be able to do anything with. And I'll come back to that in a second. Then depending on how risky it is, and each domain has some cutoffs that says whether it's a low concern, which is a 1, or a very high concern, which is a 4, we give that a score. And then for the 7 domains, so you could have something which is 7 times 0 or 7 times 4. So you could have 0 to 28, and we scale that to 100%, just simple scaling. One debate that we have to have for the biosolids use case also is if you have no data for some domain, you don't have ecological hazard data, you don't want to give the chemical a free pass for that and give it a 0 weight. So we actually set it to 0 initially. And then when we start adding up the scores to get the full score, we actually give it a 1 or a 2. And that's a judgment call we have to work through. But you get penalized on the information availability metric. So it shifts to the left on the graph we'll talk about. Next slide, please. Okay, so that's the basic approach for the scientific domain metric. Now, if we look on the information availability metric, we look at all the relevant studies that you might want to do a risk assessment. And depending on chemical properties, you may modify what's required. And so let's go to the next slide, which describes that. One more, stay here for a moment. So this is really how much of the information do you have that you would require to do a full-blown risk assessment. That's really what this is judging. And you get an overall score from 0 to 100%, but the underlying data that the users get say which kinds of data is missing. Do you not have skin irritation data, or you don't have aquatic long-term data, for instance? And the modifying criteria, which is the next slide, is an important idea. So next slide, please. So look at the table to the left. There are 15 kinds of data we would bring in. So mammalian acute, mammalian repeat dose, which is, and then specifically reproductive developmental neurotoxicity and cancer, genotoxicity, skin and eye, and so on. So, and then the eco, do we have aquatic, vertebrate, plant, invertebrate, and do we have short-term and long-term? So for most chemicals, you simply count up how many of those 15 you have and you divide by 15. So if you have an example of all of those, you get 100%. If you have half of them, you get 50%. If we now, the big modifying criteria table, you say, is there a high quality public risk assessment already for the chemical? Do we have an IRIS or a PPRTV or a ATSDR, Cal EPA? I think those are the ones we use. We assume those have done a human health risk assessment and they have found enough data in categories 1 through 7 to be satisfied they could do the risk assessment. And so even if we don't have mammalian cancer data, for instance, IRIS has decided there's enough data from their literature searching to do that. So we don't penalize lacking one of those in our public data scraping. Okay, the third column is the chemical, is it a chemical intermediate, which is really not relevant for biosolids because these are chemicals that have gotten out into the wild and it has a short environmental half-life. There we assume you would not have exposure to ecological species. And so you don't penalize things for not chemicals for not having the aquatic studies. Then the fourth column is the chemical low water solubility. Again, we assume that it's unlikely you'd have high exposure to aquatic species, so we don't penalize for those. And then finally, this is a very TSCA-specific criteria. Is it a molecular weight greater than 1,000 or an exempt polymer? There they only worry about things which have skin sensitization or very high exposure from some route that we hadn't anticipated. So anyway, you for each chemical, you put it into one of these 5 columns and you do the arithmetic to get the information availability metric. And in reality, most chemicals fell into column 1. Next slide, please. So you get a value for each chemical from 0 to 100% on the scientific domain, 0 to 100% on the information availability, and you put it in a spreadsheet with lots of columns where you make this little plot at the bottom. The next slide. And so let's look at the graph on the left. And this is this 238 chemicals that are plotted. And the most important ones to look at are the red triangles and the yellow triangles. So the proof of concept included the chemicals in the first round of the TSCA group designating chemicals as being high priority or low priority. And so we did not, the PICS process didn't know that going in. But ideally, PICS would say those high priority chemicals they should have plenty of data to do a risk assessment, and they should have high concern, so high on the y-axis. And the yellow chemicals should have relatively low concern, but they should also have high information availability. One interesting thing about TSCA is if if a chemical is designated low priority, you don't have to do a risk assessment. But if somebody out in the public finds a worrisome study, it essentially takes that chemical and pushes it into the high priority list. And so the TSCA group, it behooves them to make sure that there's good data coverage for the low priority chemicals. And there's one yellow dot, yellow triangle at the far left, which I'll look at on the next slide when I get to that, and kind of an interesting case. The right-hand graph just shows summary statistics for a variety of other inventories we looked at. And I'll just focus on the biosolids, which is that dark brown dot. It's at the bottom left. It's below the blue one. And on average, the biosolids chemicals were of low concern, but they were also of low information availability. But there are few chemicals. The whisker goes way out to 90% on the x-axis. So there are some chemicals that have very high values, and it goes relatively high on the y-axis. Some chemicals are of concern, and I'll show you the actual data points for that in a slide or two. So next slide. Slide, please. Back. There we go. Okay. So this was an interesting chemical. So the TSCA group had designated that as low priority, but we couldn't find very much public data for that. And when we into this, it turns out this was actually a combination of, they were using some CBI data, which they subsequently released, and doing some read across. So PICS does not do read across. It's all using it at, real data that we can find from an experimental study of some sort. Okay, so next slide, please. All right, here's what happens with biosolids. So the left-hand graph is the same one you've seen before, except it's added all of the TSCA chemicals. So all those little black dots are the remainder of the 30,000 TSCA chemicals. And the right-hand graph is the biosolids chemicals. And this was done with the TSCA workflow, which we may modify slightly going forward. One sort of interesting point about this is there's a trend as the information availability goes up, the risk goes up. So there's a slanty line lower left to top right, and that's sort of obvious. If you run one study on a chemical and it looks worrisome, you're likely to run more studies. Okay, but the red line is a 60%, was just kind of arbitrary for the purposes of this presentation. We're going to look at all the chemicals which are the highest risk value. And notice most of those are pretty far to the right. There's one chemical at about 30% information availability, but is above 60%. And I'll talk about that. Okay, next slide. So these are those chemicals that exceed 60%. So, all things being equal, these might be the ones prioritized for doing the biosolids risk assessments. What's interesting and actually comforting in a sense is that all but two of those, and there may be one that's maybe three, there's maybe one that's cut off at the bottom of my viewing anyway. All but two of those have public risk assessments. And these are, again, either IRIS, PPRTV, ATSDR, or California EPA. And often they have both cancer and non-cancer risk assessments. So these have already been assessed, and we have good points of departure, and we know the kinds of effects these chemicals cause. The two chemicals, N-nitrosopiperidine and P-cresol, don't have assessments that I could find anyway. And so those might be ones one would prioritize. But again, we're not looking at specific chemicals here, but this would be notionally how you would decide which ones to put into the biosolids risk assessments. The N-nitrosopiperidine only has 33% of the data. And so that would be one where, but clearly the studies that are there are bad. So they, something is happening at low doses. So one would want to go dig in and try to do some kind of a literature survey to see whether there's more data. Next slide, please. And so finally, some key takeaways. This PICS helps us understand the landscape of publicly, this is important, publicly available information for some chemical inventory. It very efficiently, I punch a button and it gives you the ones that are low and high priority candidates. It's very transparent and reproducible. Anybody can look at the data underlying it in the code. And it's broken into these modular domain-specific workflows, which can be, played with in a Lego fashion. So next slide. And so finally, in conclusion, we've done initial prioritization of the biosolids for their risk assessment. We're going to be adding in the biosolids exposure models. Okay, so that's going to be very different. We believe first pass, the scientific domains that we use for TSCA are the ones relevant to the biosolids. This information availability metric tells us how much of the information that one wants for a risk assessment is already there. And, we can rapidly run through hundreds to thousands of chemicals. The things we're doing next is, again, the exposure models. And when we did TSCA picks 3 or 4 years ago, since then we've greatly expanded the amount of data we have and our QC process. Next slide, please. And just acknowledge this was a cast of thousands worked on this project. So why don't you move back a slide? Then I don't know whether it's question time or it's time to take a break, but I'm done. Thank you very much.
Thank you, Richard. I guess I should call for any quick questions for Richard regarding what he just gave an overview of.
I have a quick question. It's Lisa.
Sure, Lisa.
Well, maybe two. So for things relative to the carcinogenicity domain and the mutagenicity, how are you ranking, 1 to 4 on those? Are you using, like, for the carcinogenicity, are you using sort of a similar scheme to IRIS, like if it's a known human carcinogen, it gets the highest rating, and if it's, only been in animals, it's, like a class B.
Exactly. That's exactly right. We mapped the IRIS and the IARC levels to the 1 to 4.
Okay. And for mutagenicity is
what we did there was we used both data, and there's some QSAR models, and I believe that case we only used 3 levels. So if there was good data where QSAR model said it's not a genotoxin, it got a 1.
Mhm.
If it was a QSAR model said positive, it got a 2. And if we had experimental data saying it was a mutagen or genotoxicant, it got a 3.
Okay. And I guess the only other question I had based on this was, that 7th domain that you mentioned, and I know that was something that was mentioned in the written comments that Shanta passed around that have been received. Though in terms of like eye irritation and skin sensitization, a lot of times those are associated with more short-term sorts of reactions and exposures, whereas for the biosolids, it seems like we're looking at an extremely long-term duration exposure interval. So I guess, is sort of the chronic versus, maybe a more acute or subchronic sort of concept. Does that factor into the domain selection at all, or?
We actually discussed exactly that question. And
Okay.
the concern is you can get, under a short-term exposure, one can become sensitized to a chemical, and that sensitization may last the rest of your life. And so we felt it would be unwise to exclude at least the sensitization piece of that up front.
Greg,
Hi, thank you. A question on whether or not the models consider the biosolids matrix at all in exposure and release mechanisms. We found, there's a lot of difference in chemical behavior when they're within the biosolids matrix, and wondered if that was considered at all.
That's going to be part of the biosolids specific exposure models. And I don't remember whether I think David's going to talk about that. So I can't really address that question.
Okay, thank you.
Ian,
I wondered if you could give me an idea of what you consider short-term and long-term.
So for differs from human health to eco, and I'm more a human health person. So short term is 28 days or less. Long term is 28 days or greater. Fish, fish and frogs, and I believe the cutoffs are much between short term and long term is much shorter, just partly because of the life scales. But I don't want to say more than I know there.
Paul,
Yeah, so first question, has the PICS process been run for the biosolids chemicals already, or you still need to do some modifications to do that?
So we ran it at the same time we did Tosca. We just put the biosolids in. And so the graphs I was showing was for the biosolids 700, where we have not rerun since we pulled in, double the amount of data we had from back then.
Right. And then
can I chime in there too on that? Sorry. There's also a list, Paul, at the end of the white paper where you can see that first attempt, how it was done for the chemicals and biosolids.
Yeah, and then the other question I had was with respect to the PICS process, exposures seems to come into it in the human health hazard to exposure ratio, or the other, the other similar equivalent factors. Is there any other place where the exposure component is considered?
It's just as part of that ratio. In principle, though, it comes in with the susceptible populations, but that's the susceptible population metric is calculated in a very different way.
Thank you.
Florence,
Yes, I wanted to ask, how does the half-life of the biosolid chemicals factor into weighting of the high/low risk? What's the criteria? What's the framework for the half-life of the identified chemicals?
Yeah, so we get the half-life, and essentially have a scoring metric that experts, and I'm not an expert in that field, said if it's short, very short, it gets a 1, a low risk. And if it's very long, it gets a 4, it's a very high risk. And there was some debate about is it an hour a day, a month, a year for those cutoffs. And in principle, we could adjust those cutoffs between the different rankings for a biosolids specific application. Somewhere in the Tosca report, those cutoffs are specified.
So short would be, you said the days, number of days, is there a specific, you said about 28 days? Did I hear it right?
No, okay, so that was a different question. For the half-life, again, I just don't remember what the exact values were. Short may have been less than a day, and long may have been greater than a year, and intermediate was somewhere in between there. The 28-day was, do we define a short-term study for the information availability where's the cutoff between short-term and long-term for the information availability metric.
Thank you.
Thank you, Richard. Well, our agenda says that we are scheduled to take a break, but if you are all up for it, maybe we should move on and do the second presentation, then have a break before moving into the discussion of each of the questions, which may take a fair bit of time as well. Are you up for plowing ahead?
Sure.
Yes.
All right. Okay, let's just keep going. Yay. All right. The next presentation is Mr. David Tobias. Are you on, David? I saw you before. There you are. Excellent.
Just give me a sec here to.
Yes, I apologize if we're catching you off guard, but
Yeah, I thought there was going to be a break there.
well, we'll take a break in a bit. After you.
Trying to make sure, hopefully everyone is going to see my first slide there.
Yes.
So again, I'd like to thank everyone on the panel. This talk is going to follow up after what Dr. Judson just presented. So this is going to focus mainly on the screening tool we've developed to evaluate the exposure pathways and compare them to hazard to get a first quantitative estimate of risk. And then we'll give a bit of a shorter section on the different ways a refined risk assessment may be carried out afterwards. So through the whole talk, just to give an outline to make sure you understand, what's going to happen here, we're going to start with, why we do what we do, the statutory requirement from the Clean Water Act. We'll have a discussion of what we think the goal of the SAB review is, the overview of the entire risk assessment framework, a little bit of background on risk and land application of biosolids, a further summary of that risk assessment framework. I'll go through one of the chemicals that came preloaded in the biosolids tool, that was aluminum, give a final summary, and then we'll move into the SAB charge questions after my presentation. So this is section 405 of the Clean Water Act, and it requires the EPA to identify toxic pollutants based on available information on their toxicity, persistence, concentration, mobility, potential for exposure. So really what that's outlining is all the steps you do in a chemical risk assessment to evaluate, as it goes on to say, which may adversely affect public health or the environment. The other thing that we're required to do under the Clean Water Act is not less than every 2 years, we should search through the available information and determine if there are further chemicals that we need to evaluate them for their concerns due to their presence in biosolids. You'll note here that it says sewage sludge. That's because the statute and the regs all refer to sewage sludge, but the biosolids term has become more common, particularly for land application. So the goal we're looking for this SAB review, as Colleen kicked off at the beginning, is that we're looking to assess human health and ecological risk from pollutants that have been found in biosolids. We're asking the SAB for feedback on our framework, and we're hoping you'll take a strategic look at that framework and how it can be implemented across a large number of chemicals. And as Colleen already alluded to, we've got multiple steps. This 3-step framework includes an initial prioritization, then a more detailed look using a screening level risk assessment and the tool that we've developed for that. And then for the chemicals that indicate they may pose the greatest risk, we'll perform a refined risk assessment. And we view this framework as supporting the EPA's goals to protect human health and the environment, consistent with what we're required to do, as I laid out in the previous slide under the Clean Water Act. And so we're seeking your comment on that overall risk assessment framework, including those 3 steps. that have been outlined now several times. So the key thing I think that you can take from Richard's talk is that PICS hopefully will allow us to rapidly look at which of those 700 chemicals need an in-depth look across exposure pathways to evaluate what doses and environmental occurrence might be to move into actual, detailed exposure calculations. And the good thing about PICS is that it taps into a large number of databases that the EPA has organized and does it in a repeatable and transparent, systematic way so that people can understand why we are moving chemicals from an initial stage to a screening level risk assessment. The next step, that screening level risk assessment, is done using the tool we've developed. It's a deterministic single point risk assessment, but it does cover a wide range of exposure pathways that may result from the presence of chemicals in biosolids. So that includes what happens when you dispose of sewage in a sewage sludge landfill, but also when you land apply biosolids, there'll be exposures from crops, meat, milk, and all of this is described in more details further down in the talk. We consider inhalation and ingestion pathways for human health. And so that includes dietary and drinking water exposures for a farm family that's living next to the field where biosolids land application is occurring. And in addition to this, we have ecological receptors that are exposed from both adjacent water bodies and terrestrial organisms, including avian species. And so on the terrestrial side, it's focusing on dietary or ingestion exposures, but then there are other organisms that are in soil or sediment or water that are exposed through the media that they inhabit. And so finally, in step 3, we'll move on to giving a general overview of what the different factors will be considered when you're doing a more refined risk assessment that hopefully will be focused more on a restricted set of pathways and receptors than everything that's presented in the Biosolids tool that we've shared with the panel and on our webpage. So just to give a very high level overview for what is risk when we say we're looking for it. So it's the chance of harmful effects to human health or ecological systems that result from exposure to an environmental stressor. We use scientific information to inform both those estimates, both the estimate of what the dose or exposure levels are, and then what hazard value should be placed for a chemical in terms of when effects might be seen due to the doses that either humans or environmental receptors are exposed to. So the exposure assessment that I'm going to describe goes through how those different doses are calculated. And if you go through the Biosolids tool, you'll see that there are multiple points where you have the opportunity to enter in hazard information. And so Richard alluded to that a bit in his talk when he pointed out that for the highest prioritized chemicals, there are what he called IRIS assessments, which is a group in ORD that does hazard assessments for chemicals, so that hazard value already exists for us to plug into the Biosolids tool. And those hazards can be parameterized based on the exposure pathway. So in the tool, there's ingestion and inhalation. Explicitly in the tool, we don't have dermal exposures right now because the framework is built around the farm family, and the assumption was that the dermal exposure route would not be the most important for them. But as Richard pointed out, there can be other scenarios where dermal exposure may occur. So throughout the talk, I'm going to speak as though there are really two kind of separate tracks for doing this. There are exposure pathways aimed at human health and exposure pathways aimed at ecological, and then different sources for considering their hazards and the number of receptors that you need to parameterize. And in some sense, you'll see that human health hazard is a bit more straightforward to parameterize in the tool, because we have right now in the tool, it's looking at chronic or long-term exposures for inhalation and ingestion. So you have either non-cancer or cancer for those two. So there's 4 data points that you can enter into the Biosolids tool when you're doing a screening level risk assessment. On the other side, for ecological receptors, there are more media through which ecological receptors may be exposed. So there are more points at which you can parameterize the tool. I think that leads back to thinking about data gaps and that information availability metric. One important thing to remember is that when you're using the screening tool while we're doing screening risk assessments is that you can run the tool without having all of the hazard information that you would potentially want to have, particularly on the eco side, as there are so many potential receptors that you could look at. The model will still run. What you'll need to do at the end is think about what the exposures in the different media indicate for the importance of that missing data, and what you may need to consider for whether, as you look across different chemicals, which ones might need extra consideration for the missing data points, or whether you need to come up with other approaches to do that. And I'll talk more about that later. So this is just a very simple introduction to use and disposal of biosolids. So there can be release to the environment from chemicals in sewage sludge when it's disposed. So if it's put in a sewage sludge monofill, the biosolids tool does have a capacity to estimate exposures that result from that. The other disposal pathway, incineration, is not currently part of the biosolids tool, but the use of biosolids is part of the tool. So that's what actually is most of what you see when you begin using the tool, is the exposures that are generated from land application. So focusing on that land application part, which is going to be central across the next several slides, is that after those biosolids are land applied, the chemicals can be transported to different media. They may end up in water, soil, sediment, and air. And so those different pathways are estimated in the tool. And for some of those pathways, they may be aggregated, particularly for ingestion to human beings, to the farm family that lives adjacent to the field. And when those are put together, they're combined with a different level of toxicity, and the output of the biosolids tool then gives you an estimate of risk across those chemicals. And you can see an evaluation or a ranking of those risks, or actually a detailed evaluation of risk across each pathway for each chemical that you've parameterized into the biosolids tool. Oh, I guess I should emphasize there that the aggregate exposure part is solely for human health risk. The terrestrial organisms do not have that same feature, nor do the other ecological organisms in the tool. So I don't want to talk too much about this. Richard already described the first step in, I think that the important thing to realize about PICS is that the reason why we need this first step is that as you start to play with the biosolids tool, you'll see that there are a lot of parameters, and a lot of those parameters, really ideally, you're going to want to put to a central tendency when you're describing their behavior, so that the exposures you get are reasonable for the farm family. But the level of work that you're doing, or the level of detail that you're putting into PICS, is lower, so you're able to run that system more rapidly across hundreds of chemicals. And also the biosolids tool generates more exposure pathways than than even in the future would be able to implement into PICS. So the PICS process may be able to combine a single exposure pathway for, say, ingestion with Richard's, the hazard values he gets from PICS. But the tool also generates inhalation exposures. It also generates, exposures for soil inverts and for terrestrial organisms and birds. And all of those different exposure pathways can't be combined simply into a large-scale process like PICS. So PICS will allow us to have some set of chemicals rise to the top. The next step is to go through the screening level risk assessment. And the important thing to remember here is that it has still some set of simplifying assumptions as compared to some full refined assessments. The calculations are based on a single value for all of the parameters, I should say, but in particular, the one to note there is the concentration of the chemical in biosolids. And that's going to play an important role in why we call it a screening level risk assessment. Using a high-end concentration is really going to cascade conservative or protective exposure values throughout the tool. Within the tool, there are several parameters that are sent to high-end values, and those mainly revolve around the behavior of the farm family itself. And there are also other ones that I'll explain a bit later that are set to central tendency values related to the transport or uptake values that describe how the chemical may move through different trophic levels or move into crops that the farm family is exposed to. And as I pointed out, if the data is available to firmly parameterize pathways and combine those with hazard values, then you'll see a risk value at the end. But if you don't have all that data for all of the receptors and exposure pathways, then you can still run the tool. And the good thing about the Biosolids tool is at the end, you get a transparent listing of all the values for the exposure values it was able to calculate combined with the hazards. And you can sort them to evaluate whether there are pathways that look like they'll need further refinement and a higher tier risk assessment. So I hope it advances, there it is, to the considering what you're going to do then after you've used the screening tool and have risks that are of concern. You're going to do a more in-depth analysis of data quality that was used for the inputs. You may want to look for further refinement of the models. The tool uses one set of models across all chemicals that are entered into it. As we'll discuss later, there are a wide set of chemicals that may be found in biosolids and that need to be reviewed under the statute. Do a further evaluation of the monitoring information and what the source of that information was and how much it may allow you to generate a range of exposures, potentially doing a probabilistic or Monte Carlo assessment. And so to be able to do that kind of probabilistic risk calculation and a higher tier assessment, it would really behoove you to have an understanding of what the distribution of that chemicals concentrations in biosolids may be. And so that would be one of the main differentiators, if you're able to do that, between the biosolids tool and a refined risk assessment, is that when you're doing a probabilistic risk assessment, you're able to incorporate distributions of concentrations or distributions of other behaviors to explore a range of potential doses or exposure concentrations that human beings or environmental receptors may have as a result of the chemicals presence in biosolids. So in the next part of the talk, I'm going to go into more depth about what that conceptual model is for the farm family and what the exposure pathways are that are calculated in the biosolids tool and considered for risk. They should line up for the most part between the screening and a higher level risk assessment for the inhalation and ingestion pathways. And you know, the conceptual model is based on subsistence farmers. They eat crops and fish from a pond that's adjacent to their farm. They're exposed from chemicals that volatilize from the field. They have exposures due to runoff and leachate to adjacent water bodies, both through drinking water and fish consumption. Aquatic organisms are exposed adjacent to that farm field, and terrestrial organisms, including livestock, are exposed via their diet. I think that one important thing to evaluate is that we feel that this conceptual model is providing high-end exposures that are relevant both for people who are farming adjacent to a field where biosolids are being applied, but also potentially to different populations that might be adjacent to a field where biosolids are applied. So if there were some other population living adjacent to a field and they were being exposed to chemicals that are volatilizing, we hope that by this screening tool, even though it isn't doing it explicitly, that it's covering protective pathways for human exposures. So if other populations would be living adjacent to a field, what the farm family is doing in the tool is living, 24/7 with adults and children, both evaluated for their exposures next to this field. If there are other situations where biosolids are land applied, whether it's parks or golf courses or wherever else land application may occur, that you've also done an assessment which hopefully gives you some confidence that you don't need to track down additional conceptual models for all of those exposure scenarios. So it's our goal that this is both applying to an agriculture scenario, but it also, because of the high-end exposures that we think it generates, covers a lot of different potential land application pathways for biosolids. Sorry. The other part of the biosolids tool and of the risk assessment framework is what it perhaps unfortunately is called in the regulation surface disposal of sewage sludge. I think maybe if people see that for the first time, it looks a bit odd. What our regulation requires us to evaluate is sewage sludge only landfilling of biosolids. So that's, I don't know, I wish they would have come up with a different term. It can confuse people because it sounds like surface spreading of biosolids, which is different. This is part of the model that you're able to access, really is more like landfilling. So you're looking at chemicals that are going to be landfilled. And when you look at that landfill exposure routes, I'll describe they're a subset of what happens when you land apply biosolids, but you have leaching into groundwater and you have potential inhalation. And when the chemical leaches into groundwater, the model considers two potential pathways, both that it could end up in drinking water and in water used for showering and bathing and result in inhalation in a bathroom. There are different liners that you have the option to evaluate in the biosolids tool, including no liner, clay liner, and a composite liner. And for that direct volatilization, again, you're getting at, hopefully what is a highly exposed population in this set. You're looking at people who live, in proximity to that landfill, so that, they have the potential for the highest groundwater and inhalation exposures. So hopefully this next slide starts to ground you in a more, visual understanding of what it is when I started listing all of those different pathways that might occur. So you have the application of biosolids to a field. In the middle of the picture there, the crops are planted. You can have uptake into produce that may result in dietary exposures. The biosolids could be applied to a pasture, and that would result in exposure to livestock and then to human exposure to meat or milk. That adjacent water body I was talking about, you see that there's the potential for exposure through fishing, but also it considers the potential exposure and risk to aquatic organisms that reside in that adjacent water body. And there are two water bodies that are actually evaluated. One is a smaller water body based on OPP's farm pond, or that's the pesticide program's farm pond. And then another one based upon a larger water body that's more of a reservoir that's thought of as being a potential source for drinking water. The other thing that the model does consider is that you could have trophic transport and magnification up trophic levels, and as well as just direct exposure from organisms that are dependent, that live in the soil and eat organisms that live in the soil. The one thing that is kind of significant but not easily depicted here is the model also considers leachate through the soil and then the motion of those chemicals into groundwater and exposure from that in the house from drinking water. For chemicals that may volatilize as well, you can have motion of the transport of the chemical into the air and then exposure to the farm family through inhalation. So this is maybe a busy diagram, but it really gives you a sense of everything that can happen, as well as on the lower right there, you see that, you assume that the farm family has access to the fields. So you could have incidental soil ingestion for the farm family, and particularly for children that may play in the field. There is a small dose that's expected to happen through hand-to-mouth behavior. So depending upon how you feel about having information presented, maybe this is overwhelming, or hopefully this complements the previous kind of pictorial representation of all the exposures are happening. But this goes through, one at a time and lays out, for each of these receptors and roots, there is a calculation done in the Biosolids tool. So you see that, after the chemicals are applied to the agricultural field, you're going to have the presence of the chemical in soil, volatilization into air, transport into surface water, and into groundwater. So this is focusing on human exposure. And so as I laid out, you have all of those different exposure doses calculated for soil, produce, beef, and milk. The model actually, you'll notice, runs them separately. It's not assumed that a family is doing both raising produce and having a dairy herd. It's one or the other that is calculated for the different scenarios that you can choose. And they have inhalation as well from volatilization that's calculated. And then from the material that's moved to the surface water or groundwater, you get drinking water, fish ingestion. And then there's the pathway that does another potential for using an inhalation hazard value would be combining it with people showering and having volatilization in the bathroom. To flip now to the other set of risk assessment, you look at what ecological exposure pathways they are. And, again, this goes back to the picture I was displaying. But depending upon which media the chemical moves in, you'll have exposure to soil organisms, plants, to livestock, to terrestrial mammals, organisms that live in the surface water, and organisms that prey on fish are also part of these pathways you see by the time you get to the receptors on the right, including plants, mammals, birds, amphibians, aquatic organisms. I would say the tool has both, it's a feature and a complication, which can be an intimidation at the beginning, and that how many different receptors are available to parameterize. I think the important thing to remember is that hopefully it's a feature for data-rich chemicals and that you can be confident you've done a full assessment. But on chemicals that aren't as data-rich as that, you still can run the tool and focus on whichever pathways you want. And hopefully, as you're using a screening tool, you're using your knowledge both of that chemical and the chemical category and of the exposure values that come up in the different media to evaluate how important those data gaps might be. So I feel like all of that got very complicated. So I try to boil it down to this much more simplified slide that gives, a basic understanding of what needs to go into the Biosolids tool. So you need to have, as I pointed out, is very important, is the chemical concentration in biosolids. And that's what's going to drive the whole risk assessment. You need to have confidence. Hopefully you have confidence that it's a high level value if you want to call your assessment a screening level risk assessment. You need to have, and then in the middle box, available chemical and toxicological properties for the chemical. And then that third box, that's more hardwired into the Biosolids tool at the screening level. That's the set of parameters and exposure factors that make this an assessment for a farm family. So it's pointing out, that they have exposures to meat, milk, vegetables, all coming from the impacted site where biosolids were applied. So, when you look at the exposure factors handbook, which is the EPA tool we use to parameterize those, many of those are set to high-end values to represent that they're getting significant dietary impact from the plants they're growing or the milk they're producing. So the Biosolids tool combines all those together to give you estimates of risk for both non-cancer and cancer endpoints for human beings, but then across all the different receptors that were depicted and described in the previous slides. So to go through it, in a little detail about what you do for the screening assessment, hopefully for a chemical, if you have sewage sludge survey data from one of the surveys that the EPA performed, you have confidence from those in terms of being able to describe what the range of concentrations in biosolids was. And we've produced an assessment of those sewage sludge surveys to extract 95th percentile concentrations. If you're using a value from our biennial reviews where we've culled data on concentrations of chemicals in biosolids, it may be a bit different to understand where on the spectrum you are compared to a sewage sludge survey. So if you're doing that evaluation, it behooves you to go and look at the literature source and try to understand what was their framework for selecting sites, how many sites were they able to evaluate, and potentially can you find further data by combining different references for situations where that's been monitored. But it is an extra complication as you look through all the chemicals we have to assess. Not only will there be a disparity in terms of available information on hazard endpoints, but there will also be a disparity in information in terms of the depth to which studies have been done on their presence in biosolids across sites. So as we move forward and think about, more of those model inputs, you're going to need to evaluate, do you feel that you have toxicological values for the receptors you need? And when you're parameterizing it, do the partitioning parameters you found allow you to properly evaluate the pathways? And then as well, you need to have understanding of the factors that parameterize bioaccumulation and uptake through the different trophic levels that are calculated in the model. So the model, it doesn't just calculate hazard and risk for the fish, but it also estimates how tissue concentrations may be building up. So that will turn into a dose for avian species. And when you're putting those parameters in, the confidence you have in this set of parameters will affect, your ability to properly label it a screening level risk assessment. And then you'll want to do further refinement if you move on to a full risk assessment. I think that when you look at these values for, in particular, bioaccumulation and uptake factors, generally what we've done when we set up the description and the user's guide is that we're trying to put them at central tendency values because we feel that the concentration in the biosolids and the behavior patterns for the farm family should already be driving conservative assessments for exposures. So in the next slide, I'm not going to go through these line by line, but what they do is they start to show what you can evaluate between a screening level assessment and a refined assessment in terms of going into more depth for each of the parameters in terms of site parameterization and those exposure factors. So in the biosolids tool, there are, everything is geared towards the farm family. The surface disposal currently allows those 3 liners. And I forgot to mention up until now, there's also a reclamation scenario in the biosolids tool. And the difference between that is this is viewed as using biosolids to make, the site again potential for, plant growth and basically introducing organic material again. These commonly happens at mining reclamation. It may happen at sites that were also, if in the past, a large amount of construction took place, there may be situations where the organic material has been removed to a point where you can no longer grow plants. So this is actually in the tool a much higher value. That's the main difference between the reclamation scenario, is that you assume they're doing a large application to rehabilitate the site. And then in the tool currently, it moves that over to after that one-time application at a high level, that it assumes you're going to have a herd on that site, and then you'd have meat or milk exposures resulting from that. So as I've described, the deterministic model is currently using 95th percentile values. You could consider that these would be varied. There's, a restricted set, 3 sites in the model that you can use for meteorological data. But if you're switching over to a refined assessment, you could implement larger meteorological databases for a larger number of sites to consider whether that would impact the exposure pathways of concern. One thing I hope you notice in the materials that we presented is that right now the screening tool is evaluating how leachate moves through the top layer of soil. And then once it moves through there, that leachate becomes what enters the groundwater through a dilution attenuation factor. And a more refined assessment where groundwater had been determined to be a pathway of concern, you could refine that by considering a vadose zone and different depths to groundwater. These are a list of just the kind of the static parameters, just to show you that there is a large amount of parameterization you need to do. Fortunately, many of these are set to be consistent with other tried and true EPA models. Like the Office of Pesticide has used the variable volume water model for a long time, and they've connected it in a large set of OPP assessments to these different water bodies that VVWM connects to. There are also set parameters for the impoundment site for surface disposal, so that when you combine the site parameterization with the other chemical properties, you're able to estimate exposures. So I'm going to switch over from describing the tool and the general risk assessment framework to give an introduction to one of the chemicals that was parameterized, and that was aluminum. I think that the chemicals we selected, hopefully you notice that they illustrate the different domains of chemicals that the Clean Water Act requires us to evaluate. So there's a drug, a metal, a PAH, and a pesticide. If you look at past sewage sludge surveys in the literature, each of those come under different potentially regulatory statutes when they're introduced to commerce. But when they end up in sewage sludge, they all need to be considered for their potential impacts on the environment. So our choice of these chemicals hopefully illustrates a range of pathways. That was the goal when we put them in. And there are citations available for each of those. When you go through the Biosolids tool, the Access database itself, each of the parameters should have a reference that tells you where to look for that. And I included the full citation that can be found in an Excel spreadsheet for all of those values. We'll focus a bit on aluminum briefly, just to give examples when you connect it to what the Access database itself looks like. One of the main, as I've described before for doing these assessments, is the chemical concentration itself in biosolids. So that value is set to the 95th percentile. The hazard values are taken from the provisional peer-reviewed toxicity values for human health. And you can go into the tool and see, where those are evaluated in the chemical properties tab. The ecological toxicity values come from aquatic life criteria. You have to, when you go to the aquatic life calculation itself, you have to choose a set of parameters beyond what the tool commonly thinks of. Because for metals, pH and the other parameters you see there will impact what the hazard value is in different water bodies. For birds and terrestrial mammals, we use hazard data from ring doves and mice, and those were extrapolated to other species that you can parameterize in the tool with body weight scaling. When you look through the tool and you're thinking about which of these parameters are going to have a significant impact on transport, the volatilization, erosion, migration, all are impacted by partitioning parameters that you need to enter in for both Henry's law constant and KD. And as these, if you were to run the model for a large set of chemicals, you would notice that those parameter values would have a significant impact on how those range of chemicals would be seen to move in the model to different media. I want to mention here that metal speciation is something that the tool is not able to track. And also the tool is not able to track degradation products for organic chemicals. So if you're concerned that a chemical you have data for also has a degradant that could be of concern, you would have to evaluate that separately. It can't be done at the same time by tracking either the degradation of the chemical you put in or the degradation of a set of other chemicals that may degrade to a chemical you're interested in. That's beyond what a screening tool will let you do. The bioaccumulation factors are also established. And so there are terrestrial and aquatic uptake parameters in the tool. If you've thought in depth about metals, you probably are aware that the concentration of the metal can impact those values, but at a screening level, there's not the ability to enter in that, more complicated behavior. So you're hopefully entering in something that allows you to get an idea for what ingestion exposures may be. But if you were to move to a refined risk assessment, you would evaluate how some of those more complicated behaviors of metals could be incorporated. I think the last thing I'm going to describe here is what does the tool at the end say for risk? And across the human health endpoints, all the hazard quotients were less than 1. There are no cancer slope factors entered, so that evaluation is not done. However, for the ecological risk, there were hazard quotients greater than 1 for the aquatic community and for some of the terrestrial receptors. So when you look in the tool, you can see the outputs for the doses and for the media to evaluate how those were compared to the hazard quotients that entered in to the chemical tabs before the model is run. So just to try to summarize all that, the EPA has developed a tool to do screening level risk assessments. And in the white paper, we describe a generalized method to look at how refined risk assessments might be done. Risk is calculated for both human and ecological receptors. And for some of those pathways, there can be aggregate exposure considered. This 3-step process is central to what we hope the SAB will consider during this review. My next slide starts to get into discussion of the charge questions, and I'm guessing we want to do that after the break. Is that what the chair thinks? I see a thumbs up there.
Yes, I was going to say, okay, let's take a break now, and then we will move into the discussion of the charge questions. And so it's I have 234 or 235 right now. So just let's take let's be back at can we be back at 240 to get going again? Quick break, and then we'll get back going again. Is that okay?
Sounds good.
Okay, quick break. Hi, Greg. I see your hands up.
Yes, I was just wondering if we'll be able to ask David a couple questions.
Yes, I can call for some open questions, but also during the clarification of the charge questions themselves, you can also ask for, further clarification on the presentation if relevant to the charge questions.
Okay.
Okay, so we are back. And Greg has so before we get into I see you're back. David, are you back? Yeah. Okay. You're there. All right. So before we get into the charge questions, David asked if you could ask a general question. So let's have some any very general questions based on the presentation itself, not necessarily the charge questions. Go ahead, Greg.
Hi, David, and thank you. First question is the same I asked Dr. Judson, just wondering if the biosolids matrix was considered in the exposure and release mechanisms in the models.
So I think that, it's set up around, so the way that you would account, I think, for biosolids specific values is that when you're doing things like plant uptake factors, or that's a good example. When you're considering plant uptake factors, you would want to hopefully start with literature where those uptake factors had been derived in situations that connect to, field studies of how chemicals in those impacted fields move into produce plants of different kinds. So that is where you would implement those kinds of things you're asking about, is when you choose parameters, to what extent are you able to tie them back to studies that were based on, biosolids land applied fields. I mean, the models themselves, it could be used for many different purposes. So it's not like the 3MRA model, which is the model for the land application site, is biosolid specific. It depends on how you pick those parameter values when you're thinking about biosolids.
Yeah, thank you. And I just would, opine that that's a big consideration. The other question, just very quickly, is whether or not the models are assuming compliance with the existing regulations. That's going to have a big impact on things like surface runoff, et cetera.
So in the model, there is a built-in, and I think there's a depiction of this in the materials in the user guide, that there has to be a set off to the water body. So there are things about how the regulations are framed that are built into the conceptual model. Sometimes it's a bit more complicated. Different classes of biosolids can be applied and then have different requirements for how long you might have to wait before you grow a different crop, if it's class A or class AEQ or class B. And you're very familiar with this, Greg, but so in terms of that, right now it is assumed that the biosolids could be applied every year. So I would say yes, in the sense that when there are regulations like distances to a water body, that has been clearly implemented into it. I'd have to think about if there were other places it would have impacted and whether those could be disentangled from the different classes of biosolids.
I think I see Ian, then Lisa. Ian, go ahead.
Yeah. So,
and then Michael.
Exposure estimates are tricky and difficult. So does the model take into account like land application farm practices, no-till versus till, biosolid method of application, injection versus slinging? How does it handle those things? And also in terms of concentration, is that total or available concentrations?
So let me start with you asked about how different application methods would impact how the chemical might move, or where it might occur. So currently in the model, in the pasture scenario, it's assumed surface application, and it gets more at that no-till scenario you're describing. And so in the biosolids, so you have that option, but with the crops, it's assumed that you're getting more incorporations. So that's the difference between those two.
That's not always the case, even for farm non-pasture crops.
So agreed, like at the level of the screening tool, you just have those two options. But if you're going to do a refined assessment, the models could allow you to consider different soil depths that, are being incorporated into the biosolids. So it would, you could consider that factor, or whether it's just all being surface applied. But within the tool, you only have two options.
Okay. And then
the pasture and the crop. And it is just a screening tool.
Whether the concentrations you're looking at are total concentrations or bioavailable.
I guess that would, you're talking about when you're loading the concentration in up front for
what concentrations are you using?
What goes in? So the concentrations you're using go back to the different methods that were used across those chemicals in the surveys. I would say, for metals, you're probably getting a total metal measurement.
That's not good.
So I think that could be good, since you're calling it a screening level assessment, that if you're able to say the metal doesn't present risks, then that gives you extra confidence in that statement. But I think that's. You have a further question on that?
Lisa, then Michael, then Jennifer.
I'll try and be brief. I have a couple big picture questions, because I think the charge questions we can get a little bit more granular in specifics. So one I have is, is the intent of the screening level assessment to really do like an MEI, like a maximally exposed individual versus a reasonable maximum exposure? Because I found, I'm very concerned about the compounding conservatism that I'm seeing, not only in the assumptions, but the setting. This person is busy, like they're not sleeping because they're, busy, like putting, doing the tilling, picking the things, eating them, cooking them, contacting soil. So it just seems to me, I'm a little concerned that we're going beyond sort of a reasonable maximum exposure. And I just wanted to understand, was that the intent of the model, or do you actually think that this is sort of an RME? And then two other quick things that go along with that is, what is EPA going to do with the output of all this assessment? So like, you have this screening assessment, I'm assuming only a subset, hopefully a small subset of chemicals might need to be carried forward into that refined, risk assessment you're talking about, which is going to be a lot of work if you're really going to try and do a probabilistic and get probability density functions for these parameters. I mean, that could be a pretty long effort, but is the goal then to use that information to maybe set limits for certain of those contaminants in the sewage sludge, and or inform like best management practices about how to apply, things to keep in mind to avoid surface flow into the pond? I guess I just want to understand like the big picture of why we're doing this and sort of how the information will be used. And I'll be quiet now.
No, I think those are good questions. I wish you would have asked them separately. I think that because you had a lot of description then of your second question. To go back, like, I think we're trying to do a reasonable high-end exposure. And I don't think that, there are other parameters. There are many parameters. So I guess you could evaluate, have we hit that for a farm family that's living adjacent to a field? And, I don't know if I agree with you that we're making them such that they never sleep. I think that, there are a range of parameters. The concentrations are set high end, but there are definitely the potential, to consider also application rates and how we've described that. And then also the range of potential, uptake and transport factors and what those lead you to for exposures. But yeah, I think that we're not going for a theoretical maximum exposure. Hopefully it's more like you're saying, a reasonable exposure. And then, to go back to, I guess your other question kind of started off with what we're going to do with these screening level outputs and how will that then inform, further consideration. I think the goal with what we get for the screening level outputs in terms of those risks is that I agree with you. Hopefully, it's a subset of the 700 chemicals such that, you're able, based on the screening level assessment, to with some confidence, given the current information, some confidence to say like, no, we've looked at these chemicals, and for the chemicals that may need further assessment, we now know a lot more about which pathways need further refinement. So it's not just everything that's in the biosolids tool. You're like, you've hopefully narrowed it with the tool to say like, this is a meat and milk issue, or there are certain pathways. But and so I think that does connect to hopefully why you're looking for high-end exposure estimates, is that you want to give people confidence that the screening level can be the endpoint, or the stopping point for the assessment of many chemicals. So that is one point of it. Like, I always try to caveat given current information, because somebody could come out with new scientific information for a chemical that we run through the screening tool. But given the current information, hopefully, if a chemical, doesn't show risks of concern, then we'd say, look, we've done our due diligence on this, and we're not going to have to do more. So that really is like what that tool needs to accomplish. And I hope it's not, staying up 24/7, but, hopefully, it is by looking at a farm family that has high-end crop and meat and milk and drinking water exposures that you're able to say like, yeah, not only like we've done it across the pathways, we've shown that this chemical is done for now.
Hey, David, it might help if you talk a little bit about how this screening model is similar to the way that we've assessed chemicals in the past, and the use of the farm family scenario in previous assessments.
That's a good point. That I think it's consistent with the public assessments or the finalized assessments that were done to support the rule, kind of the goal, and in particular, it's consistent with how the dioxin and PCB assessments were done in 2003 in terms of looking at those exposures. I think that hopefully we've actually moved in this tool to, if you looked at the 1993 assessments where the metals were first regulated, that we've moved more towards being able to say it is a reasonable high-end exposure as compared to those assessments. And so that was part of the reason why the screening tool was developed, and why informed how the 2003 assessments were done. And if you're aware, there were also some other draft assessments released over the, in the 2010s that had a similar framework. I don't know if that's actually getting to what Colleen was hoping for. You can feel free to say what you were thinking, Colleen, if I missed it. But I think that this is, trying to be a reaction to NRC feedback and feedback we've done on assessments in the past.
Michael.
Oh, okay, thank you. Thank you, David, for a very informative talk, presentation. But I wanted to ask just a general question. When we look at today's federal rule on biosolids, we identify domestic septage as biosolids and their land application. And I was wondering if the agency had considered, especially for the exposure to the farm family, the fact that they were likely to have a septic system, and it may in fact land apply domestic septage according to the rule. I don't know if that would create overly conservative risks, but I just wondered if domestic septage was part of your investigation.
So currently this is definitely focused on considering POTW sewage sludge. The available data on domestic septage is going to be a bit harder. I mean, the tool, if you have concentrations for domestic septic, for septic systems, you could implement that. The tool gives you that flexibility. And, if for chemical sets you had awareness of how that concentration may differ from POTWs, and if you were seeing significant differences, then you definitely should think about that. I think that the current regulations, one of the challenges is that, there's not a requirement for measuring chemical concentrations and domestic septage before it land applied. You have to do the pathogen and vector attraction, treatment steps, and consider, there are special descriptions for how to consider domestic septage in the rules, and you have to go through those. But it does not include gathering chemical concentration data like it does for POTWs, where they have to evaluate concentrations of the 9 regulated chemicals. So I hope I'm getting to what you were asking about, and saying that if you have the information,
the tool can be applicable if you have the information to support it. Okay.
Yeah. And you'd want to consider, would a farm of this size be able to apply 10 tons per acre of domestic septage for these number of years? That might make it, I'd have to think about whether that might make it too high end. You would have to do a bit more thought, because those parameters are kind of set up that way, although you can alter the application rate in the tool as well. So you would, I guess my point is, you would need to do some thinking, but the general models might be able to help you if you had the concentration data.
Okay, thank you.
Jennifer, Linda, then Drew.
Thank you very much. And thank you for a great presentation, David. That was really helpful. I just had 2 questions. Actually, I had the same question as Lisa with respect to the reasonable maximum and the compounding conservatism. So maybe that's something we can explore more with the charge questions, but it wasn't totally clear to me. It sounded like there might be some parameters that would be a central tendency possibly, and maybe others that were high end, but it sounds like maybe we can get into that in the actual charge questions. Does that sound right?
You could ask now as well. I'm not sure. But there are definitely parameters that are set to be high end and central tendency within the tool.
Okay. Thank you very much. And yeah, just that the notion of having every single one set to the highest or the 95th percentile is
no. Just the chemical concentration is the 95th percentile.
Okay.
Right now.
Okay, that's helpful. That wasn't totally clear to me. Which ones were at the high end versus central tendency? It did sound like there was a mix, but it wasn't clear to me how they were selected and which ones they were. That's okay. It was a lot.
The chemical concentration from the biosolids is set at 95th percentile.
Okay.
And the behavior patterns for the farm family in terms of like their food ingestion rates are set to high end from the exposure factors.
Okay.
And it's lined out in the user's guide, listed for each of those. And I forget if, it's probably commonly 90th percentile. So like in terms of like how much produce do they eat, how much milk do they drink, those things like that. Those are set to high end to reflect this subsistence farming kind of framework. But in terms of things like the BAF for the fish or the uptake factors into plants and, the transport, that kind of transport behavior, that's you're looking to set it at a central tendency value. And the application rate in terms of tons per acre is set to 10 currently, which is definitely, we don't think it's like a 90th percentile application rate. So when you combine that with concentration in biosolids, that gives you the mass per acre that's applied. So, I know when we talk to people, you could definitely have something higher than 10 if you were looking for 95th percentile tons per acre.
Okay, thank you. No, that's helpful. Go ahead, sorry.
Maybe we should stop there. There are a lot of hands.
No, that helps. That helps a lot. Thank you.
I'm trying to balance this between saying it's 3 o'clock too.
Yes, understood. I had one other high level question, which is I didn't see any dermal contact scenarios in the human health pathway matrix. And so I was just curious about that, why that is.
Because for the farm family, we're assuming that pathway is not going to be as significant as the other two, as well as, I think there's an awareness that coming up with dermal specific hazard values is more challenging as well. I think that you could argue that the dermal could be considered in other situations where biosolids are used. People may be aware that Class AEQ biosolids are sold in hardware stores. You're going to rip open the bag and apply it around your house. But if you're flinging biosolids into a field or injecting it, the assumption is that the farmer isn't out there with his hands, doing that application. So that's, that is what's built into the model right now for, not doing that at a screening level for the family.
Okay, thank you.
Linda, Drew, and then Florence. Oops. Yeah. Linda, you're muted. There you go.
Linda.
Yeah, I put my hand down, forgot to.
There she is.
Yeah, I had a similar question of Lisa's, which I want to get to in a second because I'm not sure you answered it thoroughly. But when we're again that potentially over conservatism, although you clarified some things, and when we talk about water ingestion from land application, there's a good chance, a much higher chance that leaching from that field isn't actually going into their groundwater. It moves and it might not even be their water source, right? And then the same thing with, I think why Ian asked about application type, because that really affects potential inhalation and all that, because just like with swine manure, subsurface injected to minimize the odor, which you can compare to volatilization. So, but I think you addressed a lot of that, and some of that gets granular in the higher level assessment. So, when we get to that next higher level assessment, so like when you're doing Monte Carlo simulations, where you're not just going to be thinking about the 95th percentile high chemical concentration, which I think is really important. 95 percentile is pretty high. How will that data, when you get these outputs from Monte Carlo, what's the framework for assessing that data? It might be later in your modeling thing that I haven't gotten to, but this gets back to Lisa's one of her questions is, will this data that comes out of the modeling framework, which I think it's a really good start, will it lead to both maybe limiting certain chemical concentrations? So, just like in the metals pre-treating or whatever for coming into the POTWs, or will it also, affect some of the management practices? Is that the goal, that this data will help us to better manage, the biosolids? And then also will there be this comparative thing like, okay, if we go landfill it all, the risk there versus the risk of land applying it with better management? So I just want to know, again, the big picture framework about how the hope is on how this data will be used from this multiple level screening.
Well, there was a lot there again.
I know, it's back to, it's just really how will this data be best used? Like, what is the framework for using the data?
So try to stop me if I don't answer what you're looking for. I'll do my best because you brought up a lot in your question. So in terms of thinking about conservativeness and the kind of those concerns you brought up, like, is it impacting their neighbors' drinking water or something like that?
Yeah, I think you addressed that kind of.
Okay.
Already.
Oh, sorry. Oops.
I think you kind of addressed it in when you said, well, not everything's 95 percentile and, we're trying to get the over conservative, like what can we knock out first? But now when we get to the refining, the higher risk. Yeah.
I do also just want to say that like even though some of the exposures are aggregated, they are also listed separately. So, like groundwater is listed by itself in the output of the tool. When you move to like a higher level risk assessment, I probabilistic is something that can be one of the choices, but another one of the choices could be to evaluate in depth more of some of the other assumptions and data sets you have. But I'm kind of confused by, and I don't want this to become too much a discussion of moving to risk management, but I think that, when you look at what's been done so far, there are, you're right, like it impacts pretreatment, and there may be, in the past there were concentration limits set, but also, offsets from surface water set. So it depends on the pathway and in terms of what you would move to next in terms of considering. So, you have to, I hope, like the answer there is you have to be careful to think about, what do you need to mitigate in terms of exposures, in terms of what you move towards doing a risk management action. But a risk management action would also consider more things than just the risk assessment. As you've seen, if you've looked at other EPA risk management actions. But I think when you think about what would happen in a Monte Carlo assessment, I think the hope would be that, as you would look at a broader range of exposures, perhaps you would gain, some confidence that perhaps in your initial screening level assessment, you'd gain an understanding of were you being over conservative? Does it, is that really only representing a very small percentage of what the population's risk would be? I'm not sure, what exactly you want me to say about, how a probabilistic assessment might have a next step of impact.
Yeah, well, I think you kind of addressed it. It will, how realistic was the initial risk, or how, how was that too over conservative? But maybe it also, I guess, provides, if we made a cutoff at a certain place, then we can be more comfortable at, like, maybe it's not the 95th percentile, but maybe we can be comfortable at the 60 percentile. I'm just for like a chemical concentration. So, okay.
Can I offer a comment as well? So I'm concerned that folks think that we may be regulating chemicals based on a screening assessment. That is not the case. Okay, so we're trying to develop an overarching framework for the hundreds of chemicals that we know have been found in biosolids, and move them through a screening process that's sufficiently conservative so that if there is no risk identified, we can be reasonably sure that they should be low priorities for us. The screen is intentionally designed to be somewhat conservative so that we're not missing, the chemicals that may pose a risk. This screen is also designed to be consistent with our previous risk assessments. And so we're suggesting or proposing using this similar, farm family scenario and some of the same inputs we've used in the past. If a chemical is demonstrating risk at the screening level, then we may consider moving to a more refined assessment if that's necessary. And that's when we would really dig in and find, do the distributions, the Monte Carlo, get the best data we can, do a systematic review, all of that, do an additional external peer review on that particular risk assessment. And then if there are pathways of concern following that, process, then we would move to a risk management process. So it's the risk management is separate from this process. I hope that helps a little bit.
Brew.
So David, I had a question around the terrestrial assessment, and I think you said the endpoints were dove and mice.
I said the hazard data that was used for aluminum was from doves and mice.
Okay.
And then it was read across to other receptors. You could, that's not a limitation that you need to have data on those particular species.
That was the point I was going to make, because those aren't really the best two, I wouldn't think, for assessing biosolids. But
I think that you can look at the receptors we have in the tool and think about how data availability would allow us to parameterize those. That's described in the manual.
Yeah. Okay.
And Florence.
You're muted, Florence.
Florence, there you go.
Yes, okay. So I was saying I'm going to keep mine hopefully very simple and straightforward. One of my comments or questions may be related to the previous presentation, but maybe not. So let me start with that. The susceptible population exposure is based on children. Is that right?
In the screening tool, that's the susceptible population, yes. If you did a more refined assessment, you could consider if that chemical implied the need to do other populations. And as I pointed out, hopefully by the way the framework is set up, that if biosolids land applied field, it's providing high-end estimates of exposure for multiple pathways. So hopefully that's providing some confidence that you're understanding how to put it, how hopefully that's providing protection for populations outside of just the farm family, if that makes sense.
So yeah, what I was actually getting at, in addition to the children, some of the screening tools also include in that susceptible population, the elderly, or not just the children. So is that something that might be considered?
So I think that in the screening tool, the main reason why children are an easy susceptible population is because they have ingestion rates per body mass that are clearly higher, and they have human health endpoints that those couple to, like developmental. I'm not sure how I would implement an elderly population as a an explicit calculation, but yeah, could try to consider it, but it's not in the biosolids tool. The tool just has children.
Okay. So the other question I have is in terms of you mentioned that the biosolids are one of the sections is using it for reclamation and for adding back organic matter, for example, to mine sites, post mine sites. So considering there are about 700 different potential chemicals, has there been studied that ensures that we're not putting back some of those potential chemicals back in the soil as we're using it for reclamation? I can see the organic matter addition, for reclamation, but I'm wondering the rationale for that, for reclamation.
Ooh. I think that you know, at a mining site, you would probably find. I don't know if you're worried about. I'm kind of confused. I'm worried about how you're comparing the impact, like at the reclamation site for these biosolids applications, you know, for these chemicals. I think that, you know, the goal is to show that when you're using this as a, you know, a tool for reclamation, that whatever chemical you're assessing, you know, that putting it there at those levels will not cause a concern. I don't know if that's getting to what your question is though, for reclamation sites.
Let me see if I can make my question. I guess, you know, when you're reclaiming, I really see the potential in terms of adding organic matter, trying to bring back, you know, biodiversity there. But I was just asking questions from the framework of potential hazardous chemicals in the biosolid if applied in this site for reclamation. Then the only thing I can think of now might be some microbial, you know, reclamation in the biosolid, which again, I'm not even sure because of the processing of biosolids. So I'm gonna try to get some clarification from that.
I should emphasize that is the one thing you just brought up. You know, the whole microbial risk part, that is not part of this SAB or the biosolids tool. You know, that is a separate evaluation process actually for thinking about how microbes might have impact on, you know, any of the exposure pathways. Is not what we're looking to do with this biosolids tool or whatever refined risk assessment. These are just chemical risk assessments. I think that, you know, the goal would, for, you know, the biosolids tool, for, you know, the range of materials, that range of chemicals we'll assess for reclamation, is that hopefully by you know, screening them at a high concentration compared to what they've been seen in biosolids at a high loading rate that corresponds with what happens at reclamation sites, that you're giving confidence that that won't be a concern. So I think I hope that's getting close to what you're looking for.
That's fine. Yeah, I think that's fine. And then just lastly, and again, hopefully I'm not going outside of the parameters, on the, I know the farm family has been used as the model for determining, you know, what happens at very close proximity to the biosolid they live on the site. Are there other ranges of proximity for, like, say, 5 miles away or 10 miles away, for the screening tools? Or is that something that it's been considered, not just on site, but proximity from the biosolid?
So the biosolids tool is just giving you this very near field exposure, this very near to the farm site. I think our hope is that for the pathways, that in general you would find that, you know, that these are the higher end exposures. At least when, you know, the way we're doing this is by, you know, we're considering this as kind of a conceptual model for what we need to do is address, you know, a national level risk assessment for these chemicals. So, you know, for these particular pathways, is it possible that I understand, you know, like if your question, I think, might connect a bit to what Linda Lee said when, you know, she pointed out that, you know, at particular sites, maybe it's more likely it's not actually the farmer's well, it's actually someone down gradient from the farmer.
Correct.
You know, who's being more impacted. Correct. You know, the goal of how we've structured the model, though, is that by, and, you know, using a conservative groundwater, you know, exposure model, that you're getting a high exposure dose. And so hopefully that will be protective for the farm family. And in the case where it's actually someone, 2 doors down or however far down. So that's the goal of the modeling, is that you're framing all this in terms of a farm family, but to the extent possible, when you don't have site specific data, which we can't really access when we're trying to initially think about, do these chemicals need to be regulated at a national level? You're kind of doing a generic assessment. And hopefully for each pathway, we provided protective exposures. And so I think that, like, these are things I think about, though, like you're saying, like, what about people who are, do CSAs, community supported agriculture? They're going to get a lot of potentially their diet from one farm. Well, we don't have that explicitly in the biosolids tool, but the farm family is eating a lot of the produce from their farm. So they should be protective of people who are doing CSAs. That's the goal of this framework, is to try to look at it that way, is that, like, the way it's been created are protective exposure assessments across these pathways. I think in one of the charge questions, we maybe not, I shouldn't say that, but I think that, as you're moving to a refined risk assessment, you could consider some of those things in more detail to make sure that you're doing it properly.
David, thank you. I think we need to move on to the charge questions now themselves, and remember that the goal of this part of the discussion is really to seek clarity on a question if we're not clear on either the intent or the desire to address a specific question. So the questions that the panel may have should be focused on whether or not you understand the question that is being asked of us, or whether you need clarification in order to address the question. It's not to progress further towards answering the questions. It's simply do we understand what EPA is asking of us to do? And so I am going to go through these charge questions one by one or part by part, to make sure that we have clarity on the questions. Any tweaking of the questions must be agreed upon before we adjourn. So just be mindful of that. All right, so the first question, question 1, is on prioritization. And I'm going to read the questions, and then I'll state the names. That's for people who are not able to view the screen. Does the SAB find that the application of the PICS or PICS process to the chemicals found in biosolid is sufficient to identify the chemicals that should move to a deterministic screening level risk assessment? And the assigned reviewers, the lead is Ian Pepper, and the others are McFarland, ARP, and Naruro. And are there clarifying questions on 1A? I can't see all of you, so if you have, I'm trying to keep track of hands. Okay. All right. No need for clarifying questions. Question 1B is, are there additional steps EPA should consider for implementation during the prioritization process? Oh, I need to say who's in charge. Florence, you're in charge of this one. Sorry about that. And the secondaries on this are ARP, McFarland, and Pepper. Do any of you or the rest of the panel have clarifying questions on this particular question?
I think the questions are stated fine. They're clear and straight to the point, I believe. Someone might have a different opinion.
Okay, thank you, Florence.
Sylvie, who was addressing question 1A besides myself?
Florence, Jennifer, and Michael McFarland.
Will you send out an email with these?
Yes, I believe Shante already sent one. Shante already sent one out, but we'll resend.
Okay.
All right. Are we ready to move to question 2 on the deterministic screening level risk assessment? Okay. Does the SAB find, so this is 1A, does the SAB find the selection process for models within the BST to be appropriate for the exposure pathways for a screening level? This is only for a screening level risk assessment. If not, indicate why and provide recommendations for alternative model selection criteria. The lead on this is Dr. Linda Lee, along with Kester and DeLeo. Are there clarifying questions on this? Linda, others?
I think I'm good. And it was under the chair's memo where if you go to page 4, that's where every 3 and 4 or 5, that's where
you were assigned.
Yeah. Yeah. I'm okay. I understand the question.
Okay. Anybody else have questions on this one? All right. Let's go to 2B. Are the receptors contained in the BST appropriate for a screening level risk assessment for 1, human to health, and 2, aquatic and terrestrial wildlife? If not, please indicate why and provide recommendations for alternatives. Lead is Camp, along with DeLeo, Kester, and Lee. Anybody have questions there on this one? Okay, hearing no comments, moving to 2C. Several screening parameters are set to health protective high-end values. I think we've talked a lot about some of that already, e.g. concentration of chemicals in biosolids, drinking water ingestion rates. But others are set near the central tendency for that parameter, bioaccumulation factor. Does the SAB agree that these metrics generate reasonable high-end exposure estimates appropriate for screening for 1, human health, and 2, aquatic and wildlife and terrestrial wildlife? If not, please indicate why and provide recommendations on alternatives. And the lead on that is DeLeo, along with Camp, Lee, and Samel. I think I mispronounced your name, Jennifer, again. Anybody else? Anybody have questions on that?
So Sylvia, this is Paul. I have something of a question. The BST has an awful lot of parameters, and I guess potentially would be responsible to looking at all of them. I guess that's my question. Is anything that is a parameter that is potentially variable in scope here, or are there specific ones that we should be targeting? For example, I think in David's presentation, he mentioned some specific parameters, not everything that's in the model. So just sort of a question of scope.
Okay. Sylvie, did you want to respond to that?
Well, I'm just reading the questions. I mean, I can, my interpretation is that they're asking for our overall interpretation of the parameters, but David just opened up.
I think you're encouraged to consider all
of
of the
them.
parameters that lead to exposure, doses or media concentrations.
Great. Okay. Yeah.
And
I just want to
then-
make
and then-
sure. I'd rather cover more things than miss something, I guess, you know, so.
And
Thanks, David.
okay.
Sylvia, before we move on, along with that question though, one thing I forgot to ask, has EPA done sort of a sensitivity analysis on these different pathways that could help us maybe hone in a little bit? Because like Paul said, you know, I started looking through the appendices of all the, you know, there's a lot of assumptions. So I just, you know, any input, I guess, or insights you have, David, would be great.
I think that, you know, the what you would say, you know, the strongest is the mass per acre that's applied. So that's the concentration of chemical and the application rate of biosolids, you know, and there is a discussion of the sensitivity of parameters in the user guide for the BST.
I thought it was someplace else too, but I can't remember right now.
It is in the material we provided. There's a, you know,
Okay.
a starting conversation and
Okay.
an evaluation of, you know, which parameters likely play the most importance. And I think that, you know, that hopefully that helps, but if in terms of evaluating a little bit what to look at, but I think that, yeah, we don't want to restrict in terms of, you know, if it's a parameter that's going into an exposure calculation, then it falls under this part of the charge.
Jennifer?
Yeah, I just had a question. Would the human health effects for the high-end exposure estimates also hold true for biosolids workers who would be exposed at a higher rate than, say, a farm family?
Ooh. So the biosolids tool doesn't include like the workers who are doing land application.
I mean the actual workers who are generating biosolids, say, every day for 20 years of their career.
You're thinking about,
treatment plant, right?
are you thinking about people at the POTW? Is that what you mean?
Yeah.
So yeah, I think that to evaluate their exposure, you'd need a whole new set of exposure factors to understand what their daily exposures is. It doesn't fit within, you know, the framework we're evaluating here. I mean, the hazard values could apply to those exposures, if that, which was sounded like your direct question was. So yes, if you're able to get exposures that could carry to the same types of hazard information you might use for human health in this tool.
Okay. Are we ready to move to 2D? 2D. EPA proposes to evaluate 3 locations that have different meteorological characteristics, wet, median, and dry. Are these 3 geographic scenarios appropriate for this screening level risk assessments? If not, please provide recommendations for an alternative set of locations and a rationale for selecting the locations. And the lead is Kester with Thanuro and McAvoy.
Like, I'm good on the question.
Okay, Florence, you turned off on your mic. Do you have a question?
No, I don't. I was just looking at the wet, medium, and dry, and it's kind of subjective, so I'm wondering. But that's all. But the questions are fine, but that might need some more clarification. the medium, the middle medium is what kind of sticks out to me. What exactly does that, in terms of moisture level or dryness? But the question itself is fine.
It's regard to rainfall. So that's.
Yeah, so, okay, so there are, I'm assuming there is an established criteria used to determine wet, dry, and in terms of amount of rainfall.
So yeah, within the tool, the sites have meteorological characteristics for the three, and the wet, medium, and dry correspond to regions of the country. And it was done just as an approximate assessment of, where those sites would fall along, the range of agricultural,
zones.
Yeah. So
okay.
It's and the documentation describes a little about those sites and those parameters. Sorry, not sites, regions, really. Like I said, it's not a site-specific assessment.
Gotcha. Okay.
Any further questions on D? Okay, 2E. EPA is to develop 4 scenarios for the screening level risk assessment, including specific pathways. Are the pathways for exposure simulated in the BST appropriate for a national screening level risk assessment? If not, provide recommendations on pathways of exposure EPA should consider for the screening level risk assessment. DeLeo is lead, Kester, and Camp.
I don't have questions about this question.
Okay, you don't have questions on the question. Good. Anyone else?
Or
All right, let's go to F. This is about the user guide. 2F, does the user guide describe how to use the BST for screening at an appropriate level of detail? If not, what additional information does the SAB recommend EPA add to the guide, to the user guide? And ARP is lead with Lee, McFarland, and Summit.
I'm good with this question.
Yeah, me too.
Okay. All right, then the third overall question moves to the refined risk assessment. So 3A, the white paper describes data sources EPA intends to search to support conducting a refined risk assessment. So this is in section 7.1. Are there any additional existing data sources on exposure that can be used as model inputs for Monte Carlo simulations? It could include data related to distributions describing biosolid land application rate, timing, number of applications per year, and operating life of the farm. Please provide references for these data sources. And the lead on this is McAvoy, and others are ARP, Camp, and Sawmill. Are there questions on the intent or desire for clarification on this particular question? I see heads shaking.
Yep, I'm fine.
Sylvia, I just had one quick.
Sure, Lisa, go ahead.
So one thing I was wondering is there have been, I mean, some of these are chemical specific. So I guess, is EPA interested in maybe chemical specific information regarding like leaching of, a certain constituent from a biosolid into underlying groundwater for, looking at the dilution attenuation factor? Is that something that would fall under this, or is this really more about exposure distributions and general sorts of characteristics that could apply to, most of the chemicals? Is EPA receptive, I guess, to suggestions for specific chemicals on some characteristics about mobility and fate and transport, or no?
I don't think this is meant to be addressing specific chemicals. I think if you had, how to put it, sources or a structure for what should be done, you know, once you have a chemical, like if there was a strategy that would lead to types of information that should be considered, that would be good. But I don't think it should be, you know, like for this one chemical, this is what you should do. It hopefully is, you know, strategically thinking about, you know, how this would go.
That's what I figured, but I just wanted to confirm. Thank you.
There are a series of tables in section 7 that speak to individual elements, and most of them are not chemical specific. There might be one or two, but it appears that's what the question's directed at. On like page, yeah, table 4, it looks like.
I think Paul's right, yeah.
Is everybody good on the question? Drew, you unmuted yourself. Do you have a question?
No, I'm fine.
Okay, all right. So then we're on to 3B. Are there alternative transport models that EPA should consider for this is just for the refined biosolid risk assessment. Please explain the basis for your recommendations and provide references. And the individuals here, lead is Jennifer with Ian and Drew. Are you all clear on the question?
Yep.
Yes.
Okie dokie. And then the last is 3C. Are there additional scenarios for biosolids management that the EPA should consider for refined assessments? Please explain the basis for your recommendation. Lead is Michael with Ian, with Drew, and with Florence. Are you all clear on this question?
Sylvia, I do have a question about this particular question.
Go for it.
Okay, so I want to know what's going to be most helpful, you know, to the agency going forward, because, you know, under the deterministic screening level risk assessment, we have well-defined scenarios. In this case, I don't know if new scenarios or additional scenarios would include things like land application of dewatered cake in trenches, or land application of ultra dry pellets. Is that what they're talking? Is that what the agency is looking for, or is it looking for, you know, looking at breastfeeding infants as a new receptor? I mean, when you say scenario, what I guess what is the agency looking for in terms of scenarios? Different receptors, different ways of land applying, all of the above. I just want to make sure we do something that's going to be useful in the agency's deliberations.
So I think that the question is getting at more the first part of what you're suggesting.
Okay.
Like, are there parts of land application in terms of their, you know, the methodology of that land application that needs to be, you know, that would need to be added across the, you know, chemicals, hopefully, you know, for finalizing a refined risk assessment.
So because the issue was, Florence brought up an issue earlier about elderly individuals, that would not be considered an additional scenario under the refined risk assessment. Is that correct?
It wasn't what we were, what I was originally thinking that would be added in. So the more targeted was more from a conceptual model point of view in terms of how the biosolids are being applied and managed in ways outside of what the screening tool and that is thinking of. You know,
okay, no, I understand, David. That makes sense. That makes sense. My follow-up question would be, would it also include biosolids products or materials that were made out of biosolids that are now being land applied, such as biochar? Would that be a different scenario that we need to look at as part of this?
I don't know. I wasn't thinking that biochar would fit in, but I guess I see like the way that you could argue that it should.
Okay, I just want to make
I
sure.
I guess I have to say that I hadn't thought of that one. But if you think it fits within something that we need to consider, then it would fall under this question. Is maybe how I should have responded.
Oh no, I think that's fine. You know, I'm assuming, Sylvie, that whoever's in charge of the various sections is responsible for putting together sort of the final write-up, comprehensive write-up of all the contributions. And so I want to make sure that we capture everything that the agency would benefit from in terms of our in terms of our recommendations.
Yes, your assumption there, Michael, is correct. We tagged leads to be responsible for submitting the contributions of the little work group on the particular question.
Okay.
So I get a little bit stuck here. I think that in 2B, we had a question: are the receptors contained in the BST appropriate for a screening level risk assessment for human health? And so the receptors in the human health assessment currently are children and adults, and the age of the children are actually also described in the manual. So I kind of had thought of that as being more going to what Florence had said in terms of elderly, or you could even say, like the breastfeeding infants question, I guess, would go into that. I guess I realize now there's not exactly a parallel in the refined section. But I guess if that.
Well, I didn't want to generate information that would lead to an overly conservative, set of risk values. I mean, we could look at the elderly. We could talk about breastfeeding infants. We can talk about pregnant females. But would that be valuable to the agency? Is, I guess, my question.
It's hard for me to know before I see what you would say, but I mean, I don't feel like it was what the intent of the question was. I agree that I can't say there's no potential it would benefit the agency, but I think that like if there were a strong case for whatever populations that would need to be considered, that that input could be useful.
Okay.
So I feel the need to ask Michael and others, are you okay with leaving the question as it is written now with the discussion that we've just had?
Well, I am. I think David did a great job responding. So I appreciate that.
Correct.
All right. Any other questions and comments about the charge questions themselves?
So, this is Paul. It's more procedural. Obviously, there's a group that's intended to respond to each one, but are we permitted to, or maybe they're required to deal with each one? Are all of us allowed to contribute on any of the questions? If we should.
Yes. Yes, and you know who you can contact with your
Yes.
thoughts about specific questions.
Great, thank you.
No, we just want to make sure everything gets covered, nothing gets forgotten. All of you don't flock to one question and parts don't get addressed.
Can I just throw one more thing? I think it was addressed a little while ago. Someone had asked about dermal exposure, and I believe you answered stating that inhalation and ingestion were the two upper ranked pathway methods for receptors. So is there, because of this question here, C, for additional scenarios where the dermal exposure would be included as well?
I think, David, this goes back to you on whether or not we're going back to sort of receptors, not just people, but pathways.
So I think you're talking about 3C and whether, if you're describing a new process, would that need to have a dermal assessment done? I mean, I think if you describe a new scenario and your view is that it would require the dermal pathway to be assessed, then that would be part of your basis for the recommendation and for how you view, it would need to be carried out at, to contribute to a refined assessment. So I don't want to say that you can't consider something that would introduce the dermal pathway in 3C.
Okay.
So we currently, and we did not have any requests for public comments, so which is probably fine as we're approaching the hour. Are there any further questions or comments, or especially questions, because this is your moment to ask them?
I just want to go over procedurally again. I know Shanta talked about that we can communicate via email with our subgroup people for each of these charge questions, and we should CC Shanta. And I saw that there's a deadline in a week for preliminary, sort of comments and responses to these charge questions, which obviously is not a lot of time. So are we, like, if we're, the head responder tagged for a question, should we be reaching out like today or tomorrow to our group to get their input? And, I mean, obviously we're not going to be able to do a lot of in detail sort of comment response in this kind of time period. But I guess sort of what's the expectation, Sylvie?
Well, it is a tight timeline to get initial comments in. Getting initial comments in helps us certainly frame and plan for the in-person session. Shante, you're on someplace? Maybe we can go back to the. So this is really just preliminary comments, and I think you can solicit them as fast as you can solicit them from your group, or your group can volunteer comments.
Actually, if I can chime in here for a second.
Yeah, go ahead.
Comments, I will be following up with the panel to provide guidance on that, but everyone should be submitting those responses to me. I will then provide a compilation of those, which we will utilize at the May meeting. Leads do not need to reach out to team members at this point in time. Everyone should just be focusing on responding to their assigned questions. And if you wish to provide comment on non-assigned questions, those are welcomed.
Thanks, Shanta. So are we clear? Because
Yes.
Yeah.
Sorry, could you resend the assignment of the?
Yes, I will
charge
be
questions with them?
resending the charge question assignments to the group.
Thank you.
And I'm sorry, I do have a question, just what you just said, Shanta. So the lead for a question should not reach out to the others on that team?
Not at this point. Your lead writer responsibilities will kick in May
Okay.
for that May meeting. Okay. Feel free to capture the notes from today's meeting, some of the conversations that may be helpful for the write-up going forward. But at this point, we're just focusing on everyone providing their preliminary comments, and that will be of an aid for the May meeting.
So we should provide those on all of the charge questions, not necessarily one.
I'm asking specifically for those questions you are assigned to, but if you want to provide comment on others, you are welcome to.
Okay, thank you.
You're welcome.
And Shanta, so then if groups wanted to get together ahead of the May meeting to discuss their particular questions, they can do that, right?
You are welcome to communicate as a small group only. Just make sure I'm involved on any and all email communications. And if you have any voice conversations or meetings, make sure that I'm CC'd on those invitations.
And then
I do want to stress that this is preliminary work. Everything that ends up in the report has to be discussed at the deliberative meeting. So again, May will be critical.
Okay.
And Shanta, more directions and, sort of draft agendas for the May meeting and things like that. They will be coming out quite soon, and that will help clarify both what will happen at the meeting, but also lead up to the meeting.
That's correct. And this is for the public's information as well. We will be updating the SAB website with all of the information for the May meeting. So look out for that. Hopefully having it posted within a week or so. And for the panel, I'll definitely be following up with you by several emails.
Any final questions? Otherwise, I'm going to turn it back over to Shantae.
All righty. Well, I want to thank members of the public for your attendance at today's meeting. EPA, we want to thank you for your presentations and your response to questions. To our Biosolids panel, thank you for your participation and your eagerness and interest in this topic. I look forward to having a fruitful discussion in May. As a reminder, we do have 2 additional meetings for this panel. The first will be May 2nd and 3rd. That will be in person. Follow-up information will be available on the SAB website to indicate the location as well as the registration information for members of the public who are interested in attending. To the panel, we've discussed this, but once again, preliminary comments will be due on April 12th. I will be sending out a follow-up email to provide additional guidance on that. And with that, are there any questions?
Linda?
Yes,
I have a
Linda?
question. So I don't think we've actually decided on the times for May 2nd and 3rd. And so
those meetings will be full day meetings. We will run 9 to 4.
Okay.
Yes.
Both days?
Both days. Any other questions?
Should I stay on, Shanta, to go over my specific issue, or should I email you and we can talk after?
We'll follow up by email. Okay, with that, thank you, everyone. This meeting is adjourned. Have a great afternoon.
Thank you.
Thank you.
Bye.
Thank you, everyone.
Bye.